Hello and welcome to Chemspec Digital 2021. I have the pleasure to introduce the next presentation in the Pharma Lecture Theatre. However, before I do, please note that we are recording the presentation to make it available to view on demand. Attendees are invited to write questions into the chat function, from where we will read them out at the end. And now, I will hand over to Mark Wellman of BIOVECTRA, who will be presenting challenges and opportunities in high-potency active pharmaceutical ingredient manufacturing. Over to you, Mark.
Thank you. Good morning, everyone. My name is Mark Wellman, and I am the Vice President and General Manager for the Complex Chemistry Division of BioVectra. My role includes leadership for our operations teams at our manufacturing sites that specialize in organic synthesis of small molecules and also fermentation of small molecule metabolites. I have about 25 years of experience in the industry. So it's my great pleasure to speak with you today. And the topic of my presentation is going to be challenges and opportunities in high potent active pharmaceutical ingredient manufacturing. But before I get into the details of today's topic, I'm going to spend just a brief few moments to introduce you to BioVectra. So a little bit about BioVectra. We were founded in 1970, so we're approximately 50 years old as an organization. And we are currently a portfolio company of HIG Capital. BioVectra's focus is supporting clients in pharmaceutical and biotechnology development. And what we really care about is solving complex problems in small molecules and biologics. So you can see geographically here, we're on the northeast coast of North America. We're in the Canadian province of Prince Edward Island with facilities in the province of Nova Scotia as well. So that puts us approximately 10 hours northeast of Boston by car, and also accessible through major hubs in Toronto, Montreal, and New York. A little bit more of our locations. We have 5 GMP facilities located within Atlantic Canada. Our team members is about 500 employees, and we have a strong regulatory track record with Health Canada, the FDA, and also the Japanese Ministry of Health, supporting regulatory filings such as ANDAs, DMFs, VMFs, and CMCs. A little bit of deeper dive into our facilities. Over the 5 facilities that I just described, we have approximately 225,000 square feet of manufacturing space, about 118,000 liters of chemical reactor space, and 64,000 liters of fermentation bioreactor capacity. Now that fermentation bioreactor capacity is roughly equally split amongst fermentation vessels that are dedicated to the generation of small molecules, and the other half is focused on large molecule biologics. Accompanying that is about 70,000 liters of buffer solution prep space. So to summarize, we view ourselves as a full service CDMO with complementary capabilities. So some of the capabilities that we feature is, of course, complex chemistry, fermentation, small molecules, large molecule biologics, bioreagents, controlled substances. And then, of course, the accompanying services, analytical development, process development, formulation development, and regulatory filings. So we view ourselves as an end-to-end strategic partner from R&D through to pilot plant and into commercial supply. But one of the things that I want to talk about specifically in our capabilities with you today is high potency API and BioVectra's experience in the manufacture of high potency APIs. So to get into a detailed overview of the talk that I'm going to share with you today, we'll be looking at the history of high potency APIs, and we'll also dive into the criticality of understanding your high potency API. We'll talk about cleaning and decontamination, and we'll also touch on a word about personal protective equipment, or PPE. And then we'll roll through product considerations, scale up, some manufacturing designs, and then ongoing processing. So to start things out, we'll look at a little bit of the history of high potency APIs. And of course, high potent active pharmaceutical ingredients require very little substance to elicit a sufficient effect. Now, some historical categories include sex hormones, some opiates, antibiotics, cytotoxics, and prostaglandins. But high potent products are also often of natural origin. And these natural origin products are often tapped as payloads to form antibody drug conjugates. Now, some examples in the industry are clindamycin, which is an anti-tumor antibiotic that was isolated in the Texas chalk pits. Another common example is ensamitocin, which is a potent anti-cancer antibiotic that is expressed through microbial fermentation. And then a third example here on this page is delastin 10, which is a cytotoxic pentapeptide that was discovered in sea slugs and serves as the parent compound for auristatin, which is a potent anti-cancer payload that is delivered via an antibody drug conjugate. If you look at high potent API pharma trends, what we've seen at BioVector is clients are coming to us with drugs that are increasingly more potent. And this is a result of the scientific community able to conduct high throughput screening. So there's more and more products being evaluated. Now as well, we're also able to chemically alter the products for even activity at even more lower doses. And then tagged on to that, we have optimizations for increased bioavailability, and then also targeted drugs for specific pharmacological activity. So the results of all this great scientific innovation is that products can now be administered at micrograms per dose. But consequently, the challenge of that and the opportunity is that these products require controls and systems to protect employees, the facilities, and the environment. So one of the first things that you need to do when you're taking on a high potent API project is you really need to invest the time to understand your high potent API. So before you start, invest the time to look at your product, know your product, understand your product. Now, some novel compounds may have limited toxicity data. As I mentioned on the previous slide, the result of high put screening puts a lot of products through for consideration. And you may not have all of the toxicity data that you would require for your product before you start. So you need to consider other toxicological assessments without the full gamut of toxicity data. So you can do this by comparison to other compounds that are similar to it in the literature and applying scientific calculations that are known in the industry to generate a toxicity report on your compound. Now, once you have the toxicological data and the report on your compound, you establish banding ranges and preset protocols to ensure the safety of your employees, the workplaces, and the environment. Now, here at BioVector, we've established 4 banding protocols, which is at 0.5 milligrams per cubic meter, which is the lowest banding rating, down to band rating 4, which is 30 nanograms per cubic meter or less. And these are all expressed as an occupational exposure limit based on an 8-hour time-weighted average for an employee working 40 hours a week for over a working lifetime. You also need to consider routes of exposure. So the most common route of exposure, and consequently the most difficult to control, is inhalation. Now, inhalation generally occurs through static solids that become airborne, crystalline powders that become airborne, but it can also occur through dissolved solids. Now, dissolved solids are generally a good technique to avoid airborne generation of material. However, solutions can become airborne through aerosolization or through high pressure atomization. So you need to be careful about inhalation. Another route of entry is dermal absorption. And this can be variable depending on the chemical, but it's also enhanced through the presence of organic solvents. And then a third one is ingestion. Now, ingestion is rare and it's quite often accidental, but ingestion is generally through just common habits that we have, contamination of tools, pens, gloves, and then touching your face. And very similar to ingestion is an inadvertent contact with the skin and mucous membrane. So again, this is rare and it's generally through the same routes and the habits as we have through ingestion. So if we look at the visual limits, 50 to 100 microgram per cubic meter is the visual limit which solids can be observed in the air. So if you crawl on the previous page, band 3, band 4, we're down into the nanograms per cubic meter. So certainly you are not going to be able to see the compound at the containment level required at a band 4 rating. And the visual limit on a surface is about 1 to 5 microgram per cubic centimeter. And that's on with optimal conditions, looking at product on a surface. So the next step as you go through your APIs, once you understand your API and you classified your API into a banding range, you need to look at cleaning and decontamination. And this is a very important prerequisite to your production. So you need to develop cleaning methods that are effective at cleaning your product. And you need to also consider the ability to clean different surfaces. So not only do you have to clean your vessels, your isolators, you also need to clean surfaces within your room. You may be looking at stainless steel, you may be looking at glass, you may be looking at wall surfaces, floor surfaces, you may be looking at vertical surfaces versus horizontal surfaces. So it's a careful consideration in your cleaning methods in what you're going to be cleaning, and make sure that your cleaning method is effective against all these surfaces and situations. And then going along with your cleaning methods, you also have to develop test methods with appropriate limits of detection. So we are going down to very low limits of detection. And quite often you're looking at HPLC, ELISA, UV, LCMS, GCMS, et cetera. Now, once you've cleaned your product and you verified it has been removed with an appropriate test method, Biovectra has a standard practice of further decontamination and destruction of the molecule. So decontamination and destruction is usually with acids, bases, or strong oxidizers, sodium hypochlorite, et cetera. Now also you have to understand your waste handling methods and your waste destruction. So your waste handling can be quite often handled by a third-party contractor, and you need to ensure that your waste is thoroughly decontaminated and destroyed from any high-potent API prior to handing off to your contractors. Now, all of this is also accompanied with a strong industrial hygiene program. So you have to have industrial hygiene methods that are able to monitor the levels of high-potent API that might be in your manufacturing environment and any adjacent environments surrounding your manufacturing environment. And that's not only in the air, but also on surfaces. So our industrial hygienists will monitor air, surfaces, and personnel. They will track movements, and they will track out from the center area of manufacture out through a perimeter around the manufacturing area to ensure that our containment controls remain effective. Now, why is cleaning and decontamination so very important? And the reason for that is even the best designed equipment and process will require some maintenance. So at some point during your process, you are going to need to do a line break. You may need to calibrate a piece of equipment. You may need to replace a piece of equipment or do some preventative maintenance on an equipment. And you need to ensure that you've adequately cleaned and cleared that piece of equipment or that instrument before you allow any operations to occur. So it's very, very important to have these robust, validated cleaning methods designed ahead of getting into your process. Now, the second thing that you need to consider is containment. And we're going to talk a little bit about personal protective equipment. Now, first of all, the process and safety is contained first by engineering design, by engineering controls. So containment is validated and ensured by appropriate equipment selection. So you look at your process and you select and validate equipment that will contain the product. And we validate that containment using surrogates to study the process. So some of the surrogates that we'll commonly use are naproxen sodium, lactose, and mannitol. Now what we do is we put the equipment through the process using these surrogates to mimic the exact process that we are going to be challenging this equipment with. And we conduct industrial hygiene monitoring to ensure that the equipment selection is appropriate and it's validated for the containment level required. So all this being said, Containment is done by design of the process. It's done by design of the equipment. At BioVectra, PPE will always be worn, but it will never be used as a primary means of employee protection. It's the equipment design, the containment design that protects the employee, the facilities, and the environment. So some product design considerations. So what you need to do is, as you're scaling up your process, you need to implement both quality and containment by design. So you hear a lot about quality by design, which is extremely important. But when you're dealing with a high potent API, you also need to consider containment by design. And you need to understand the scale of your manufacture. If you're in a small scale operations, strategy that is great is to put all of your chemical reactor equipment inside of glove box isolators or containment devices. And that can be a great strategy. But what about scale up? What happens when you outgrow your small scale manufacture? So you have to look at your process. You have to challenge your process, how it's designed, and then adapt that process that's optimized into containment devices to make sure it'll work. So what we say is we want to be proactive. We want to look at our process. We want to challenge our process. We want to optimize our process. And then we want to be proactive with a good process hazard analysis, looking at the equipment hazard analysis, as well as looking at containment hazard analysis, which is very important. So looking at some manufacturing designs here, this is a processing area that BioVectra actually uses. This is one of our potent containment suites. And what we see here at the center of the drawing is our processing area. And the processing area is one of the most negative areas in the suite, of course. So all of the air is brought into that area and exhausted through that area, and that prevents any of the products from potentially coming out of the area. So all of the equipment in this area is qualified process equipment for the containment level required. You of course need to have dedicated HVAC for this area. And we also have bag in, bag out HEPAs. So we'll have HEPAs for the air intake, and we'll also have HEPAs for the air outlets. And this is to protect the employees working on the HVAC system should it need to occur. Of course, you're going to need to have airlocks surrounding to protect your differential pressures, and of course that cascade of differential pressures. And if you look closely at this drawing, you can see the cascade of differential pressures depicted here. There's separate utility and maintenance access. So the center area is the processing area, but what you see along the perimeter of these processing areas is utility areas. And that's designed so that the maintenance personnel do not need to enter the manufacturing area in order to conduct metrology or preventative maintenance events. Now, personnel entrance is through a dedicated airlock, as well as personnel exit is done through a separate airlock. Personnel airlock is done through a misting shower, and the misting shower is designed to just protect the employee by coating them with a light misting of water, such that any potential airborne particulate that may be on them is coated onto their personal protective equipment, so they are not exposed when they are degowning. As they step through the misting shower, there's a series of protocol steps that the employee will follow to degown, and also special consideration on waste handling of their used personal protective equipment as they're exiting. We have a separate materials airlock as well. But even with the materials airlock, one of the things that you need to consider is minimizing the amount of materials that flow in and out of the materials airlock. And in this case, the design is to bring all materials in through material transfer systems so that we're avoiding physical movement of materials in and out as much as possible. And that also ensures containment of the process that is being manufactured. So, and finally, there's strict access control, of course, and personnel flow into these areas. So this is all pre-designed, pre-understood. There is this accreditation for employees to be able to enter the potent compound area, and it's strictly controlled and monitored through industrial hygiene monitoring. So I hope in this brief amount of time I've been able to give you a good overview of BioVectra's experience with high potent API manufacturing. It's been brief, But just to leave you with some considerations, it's very important to understand your high potent API. It's very important to look at cleaning and decontamination. And it's very important to consider the effective design of your process, and as well as validate containment. So containment is done by engineering controls and not reliant on personal protective equipment. So at Biovectra, we have experience with this. We have 2 suites with occupational exposure limits of less than 20 nanograms per cubic meter. Of course, the exit and entry air locks, rated class 100,000 modular rooms, the industrial hygiene programs, and reactors that are capable of conducting high potent API manufacturing, isolators, enclosed biotage chromatography systems, custom enclosed fraction collection systems, isolators, filter dryers, and we're also able to conduct high potent API chemical conversion. So all of this said, we have experience with about 30 kilograms per year of band for high potent API. So with that, I'm going to conclude the talk today. And I will thank you for your time, and I will turn it over to the moderator for any questions.
Thank you very much for that, Mark. A few questions came in. I just read them out to you.
Sure.
First question, have regulatory agencies ever been concerned with manufacturing HPAPIs at your facility?
That's a great question. Thanks, Stephanie. So the answer is yes. Regulatory agencies have had a look at our high potent API containment, as well as our clients as well. So it's really important to have a risk assessment done on the containment of your high potent API and have that considered in your design and in your ongoing manufacture of high potent APIs. So in this case, we have a risk assessment study done on the containment and controls that are being used. We have the validation of our equipment that ensures containment, and we have the ongoing monitoring that ensures containment as well. So with a full risk assessment package, we were able to generate the confidence of both regulatory agencies and other clients to ensure that our high potent APIs are being fully contained.
That's great, thank you. Next question, are employees ever appreciative about working with HPAPIs?
Yeah, okay. So yes, I mean, the employees, it's always really important to engage the employees when you're manufacturing high potent APIs. And the philosophy and the culture that we have at Biovector is that we engage the employees throughout the life cycle of the product, because they are going to be involved ultimately in the manufacture of it. So when we are into the qualification of the equipment, we will engage not only our engineering department, but we will engage the production technicians that are going to be working front and center on manufacture of the product, so they understand the containment levels that are required to manufacture the product. And they're also involved in the surrogate studies. They are confident in the containment level that's being adhered to the product, and we're fully transparent with the industrial hygiene monitoring that occurs as well. So we fully involve all of our employees, all of our staff in the manufacture of high potent APIs. It's a cultural shift. Everybody understands the importance of it. Everybody adheres to the protocols that are required for the management of high potent APIs. And consequently, you have a successful workforce that is all working in the same direction to ensure that our high potent APIs are manufactured with the best quality and, of course, the requisite containment to ensure the safety of our workforce.
Thank you, Mark. And I have one more question for you. What is the most difficult operation to contain?
Okay, that's a good question. So the most difficult operation to contain is powders handling. So I mentioned briefly in the course of the presentation that powder handling is the most difficult to control. Now, if you're looking at optimal conditions, when you may see dust in a strong beam of sunlight in a room, that you're at the micrograms per cubic meter dose under those optimal conditions. So what you could see as a person with your eyes. Now, when you're down to containment level 4 or a band 4 containment, you're down to nanograms per cubic meter. Certainly, you're never going to see that with the human eye. And it's very easy to generate dust. And most of the dust in airborne particulate is generated during the final packaging of the product. So of course, final packaging is done in a glove box isolator that is rated for that packaging. And Biovector also has a protocol of overlapping with 3 layers of protection during that final packaging. So we will package in a glove box isolator because it is the most important part of the containment protocol. We will clean and decontaminate the outer portion of that primary packaging. We will overlap with the secondary packaging, and then we will overlap with the tertiary packaging as well. So that ensures the containment of the product, the protection of the product from our employees, but we also understand that this product is ultimately going to be delivered off to our client. So we ensure there's 3 layers of protection over top of the product as it's delivered to our clients.
Very good. Thank you for that, Mark. This marks the end of the session. Thank you very much to our presenter and to all of you for watching. Goodbye.